Hi everyone. Today I'm going to be talking about lipid digestion, which is the second lecture for chapter 5. As a reminder, I had surgery yesterday and I'm feeling a bit under the weather, so you will not be seeing my face on video today. Just in case you think that there's something wrong with your computer, I wanted to clarify that. What happens when we eat fat? Well, we know the process is different. The digestion, absorption, and transport is much different than that with carbohydrate or protein. Fats essentially reach the stomach intact. There's minimal digestion that goes on in the mouth. And in the stomach, the fat is broken down into droplets or emulsified. And they float on top, if you can picture this, floating on top of the digestive juices in your stomach until they're metered out and passed on into the small intestine. So a 30,000 foot view of what happens with lipid digestion and what organs are involved. As I said, minor fat digestion occurs in the stomach. There's some emulsification going on. The liver produces bile, which I think everyone knows, and that is what helps emulsify that fat. And of course, that's stored in the gallbladder, the bile, if you still have your gallbladder. And the pancreas secretes enzyme lipase into the small intestine to further break down that fat and some of those bonds. And the small intestine is the main site for lipid digestion and absorption. And less than 5% of the fat, or undigested fat, less than 5% should be present in the feces. If there's more than that, there's some type of fat malabsorption going on, which is never a good thing, believe it or not. So we know that fat enters the small intestine, and then cholecystokinin triggers the release of enzymes. The gallbladder is going to contract and release bile. And we know that it's produced in the liver, and what we want to realize is that the bile contains these bile salts that essentially work like a soap, and again breaking up that fat into smaller droplets and emulsifying it so it can be transported into the small intestine. Pancreatic lipase breaks down triglycerides into 2 free fatty acids and 1 monoglyceride. So you have 2 free fatty acids floating around and 1 monoglyceride. Phospholipase removes the 2 fatty acids that are hooked onto the phospholipid and then just leaves that phosphate group on the glycerol backbone. The monoglycerides that we just talked about and the 2 free fatty acids get transported by micelles, and it gets transported right to the wall of the small intestine. And there's a picture for you so you can see what's going on with the micelles. And the fatty acids and monoglycerides remember, they're not soluble in water, so they have to be surrounded by phospholipids, because the phospholipids have that hydrophilic tail, head that's pointed outward. So once in the intestinal walls, the short chain fatty acids can pass directly across the intestinal cell membrane. And just as a reminder, they're short chain fatty acids, and they have less than 6 carbons, which is why they can be absorbed directly across that cell membrane in the enterocytes. The longer chain fatty acids, so the 14 plus carbon containing fatty acids, have to have a special carrier protein, and then they can be absorbed. And the chylomicron is what we use as a transport vehicle to get that lipoprotein, that specific lipoprotein moved to where we want it to go. And the chylomicrons pass through the lymph lacteals. And ultimately, chylomicrons are broken down by lipoprotein lipase, LPL. This is a picture of a chylomicron, just so you can get a visual. We'll talk about the apolipoproteins in the next lecture, but you can see that the phospholipid heads, which are hydrophilic, meaning that they don't mind the water, they don't mind floating around in that aqueous environment. However, you can see the lipid tails that do in fact not like that. And then they're pointed inward. And in the middle of the chylomicron is the core containing cholesterol esters and triacylglycerols, or triglycerides, however you prefer to say it. This is a diagram from your book, and I won't read it to you, but it essentially traces a path of when dietary lipids are first ingested all the way to absorption. Again, take note that short and medium chain fatty acids don't get incorporated into the micelles for absorption into the intestinal cells. And I'll let you review that on your own versus read through it. The micelles that I was just mentioning interact at the brush border, and the lipid that they contain diffuses out into the enterocytes, so into the small intestine cells. And these lipids will ultimately end up being reassembled eventually by esterification and released into the circulation once again. So how exactly these lipids move across the brush border isn't fully understood. There's two theories. There's a protein-independent diffusion model and a protein-dependent diffusion model. So in one model, there's a carrier protein, and in the other model, there is not. And CD36, though, is something that they've identified, that a protein they've identified that does, in fact, transport fatty acids into the enterocytes or the small intestine cells. So what are the fates of the lipids that we consume or that we make in the body? Well, we need them for energy. We've talked about that. If you're stranded on that island, you're going to use that as a fuel source for a while. We have compounds in the body like steroids that are comprised of lipids, hormones, cell membranes. Cell membranes are lipids. And they also play a role in inflammatory response elements. And they can be stored for later use. Again, that's our fuel source. Infinite capacity, as I was talking about in the last lecture. We have these programs, the 800-pound man or woman, which is, I'm simply talking about to illustrate the point that that adipose tissue, that is the actual cells, have an infinite capacity for growth. And active individuals tend to actually store fat in their muscle tissue first, which I think is interesting, because if they've got to get up and go, they've got to have a ready source of energy available to them. So, as I was saying, active individuals tend to store fat in their skeletal muscle. It's readily used. If they're very active, they can get up and go, run for their life, whatever they need to do. And they actually store less of that in adipose tissue. So those fat cells actually have less fat in them. It's stored instead in the muscle tissue to be utilized when it's needed. I apologize. Let me fix that screen. I've gone over this. This is just an overview of how triglycerides are digested. And again, minimal digestion in the mouth, minor amount. The stomach serves primarily as a place for emulsification to take place. Gastric lipase comes into play in the stomach. Lingual lipase in the mouth. I'm going backwards, sorry about that. But again, it's really minimal, and a lot of resources don't even consider any digestion occurring in the mouth. And as we talked about, the primary place of digestion is in the small intestine. And I have gone to the next slide. I mentioned that triglycerides were primarily used as an energy source, and that's what's found in an adipose cell, these large triglyceride droplets, like the picture I have here for you. You can see the fat reservoir, and most of us want that fat reservoir to shrink. And the triglycerides are broken down into fatty acids in these glycerol molecules, and then they're released into the bloodstream. And cells will use those fatty acids as they're circulating for energy. And we did talk about gluconeogenesis, where the liver can convert the glycerol to glucose for another energy source in case your glycogen stores get depleted. So how much fat should we eat? We know that fat is an essential part of anyone's diet, and it's important to consume both omega-3 and omega-6 fatty acids. The range of recommended DRI range for fat in terms of calories per day is 20 to 35% of daily calorie intake. And believe it or not, most people do eat in this range, but it's just that they're not consuming the right types of fats, the heart-healthy types of fats. And recommendations are that you aim for less than 7% of saturated fat and as little as possible of the trans fatty acids that we talked about in the prior lecture. So if you have a 2,000 calorie diet, approximately 400 to 700 calories or 45 to 77 grams of fat should be consumed based on the DRI recommendation. And again, less than 7% of that needs to come from saturated fat, or should come from saturated fat. And that concludes the second lecture on your lipid chapter, and the next lecture will be on lipoproteins. I will see you, or I will talk to you soon.